Hello and welcome to this webinar titled Material Understanding for Continuous Manufacturing of Solid Dosage Forms, hosted by Biopharma Asia magazine and presented by Dr. Yifan Wang, Regulatory Research Scientist, FDA, CDER, and Mr. Dirk Leister, Head of the Application Team at Thermo Fisher Scientific. My name is Stephen Edwards and I will be the moderator. Now, please allow me to introduce our first presenter, Dr. Yifan Wang. Dr. Yifan Wang is a regulatory research scientist at the FDA CDER's Office of Testing and Research, Division of Pharmaceutical Quality Research. She serves as a technical expert for solid dosage forms using both batch and continuous manufacturing technologies. She is a member of the Advanced Manufacturing Science team to expand the continuous manufacturing knowledge base. Dr. Wang is certified by the American Society for Quality as a Six Sigma Black Belt, as well as a pharmaceutical GMP professional. With her expertise in advanced data analytics, Dr. Wang has worked with continuous manufacturing in various aspects, such as material characterization, formulation development, process optimization, and real-time release testing. Dr. Wang received her BS in Pharmaceutical Engineering from Dalian University of Technology, China, and a PhD in Chemical Engineering from Rutgers University, US. She also received certificates of completion in chemical technology and biotechnology from the Royal Institute of Technology, Sweden. I will now hand over to Dr. Wang. Welcome, Dr. Wang.
Thank you, Stephen, for the introduction. I also like to thank Biopharma Asia to give me this opportunity to work with Dirk and make this webinar happen. It's nice and sunny here in White Oak, Maryland, and I know that we're having more than 130 people joining us for this webinar from all over the world. And I feel really happy that our same interest can bring us together at this moment. Continuous manufacturing has been on the spotlight for more than a decade now, and if we compare what we know about continuous manufacturing to where we started 10 years ago, we must be very proud of witnessing the learning curve we developed together in the community. As a research scientist, as well as a process and facilities assessor, I would like to highlight some of the work going on at FDA in an effort to better understand material properties for continuous manufacturing of solid dosage forms, and also to prepare our readiness for the adoption of this technology. Before I start, please allow me to read this disclaimer. This presentation reflects the views of the authors, and they don't necessarily represent the FDA's views or policies. I like to use word cloud to catch the keywords for a certain topic. And if I do that for continuous manufacturing, we can clearly see what is commonly discussed. For example, process understanding, batch size, control strategy. And for the next 20 minutes or so, I would like to focus on the keyword that lies on the very left, the materials, especially the properties of the materials for continuous manufacturing. I will highlight the trends in the continuous manufacturing and discuss what are the material considerations for CM. And also, I would like to share with you some of the recent work using FDA's laboratory capabilities in the Division of Product Quality Research to better understand material properties for CM processes. I will then end my presentation with some thoughts on the steps ahead. Over the past decades, we have seen a lot of efforts from academia and industry to move the pharmaceutical industry towards the continuous manufacturing. Right now, we have more than 4 approved drugs that have adopted continuous manufacturing, and there has been a growing interest in implementing continuous manufacturing for pharmaceutical products through requests and interactions with the FDA Emerging Technology team. As of February this year, there have been 32 requests and 60 interactions with the FDA Emerging Technology team, and a proportion of them are related to developing continuous manufacturing platform. Implementing continuous manufacturing includes many aspects. from material properties, process understanding, and control strategy. Since I will be focusing on the material properties today, let's start with this question. What is the role of understanding material properties in general pharmaceutical development efforts? Whether in batch processing or continuous manufacturing, the bottom line here is that we don't want the quality of the final product be impacted. And statistically speaking, the variations of the product can come from the variations of the materials, including both APIs and excipients, the variations of the processes, as well as the interactions between the processes and materials. And in other words, material understanding and process understanding are usually complementary to each other, and both can help to control the quality of the final product. And through a QBD perspective, in order to explore the design space, how critical material attributes, critical process parameters affect the quality of the final product needs to be evaluated, and the material flow properties, in some cases, can be considered critical material attributes. There may also be cases where certain predictive models, such as flowsheet modeling, need to include certain material properties. And also, the ICHQ-8 suggests that the manufacturability of the drug product should be identified and discussed. And the manufacturability is always linked with the interactions between the materials and the process. And also, characterization of incoming materials helps to evaluate potential failure modes during the process. For example, agglomeration, segregation, or poor control of the weight and content uniformity. And when material manufacturability can be characterized under more process-relevant conditions, potential failure modes can be avoided. And lastly, material flow properties can also play a role in the raw material control. The Q10 defines the state of control as a condition in which the set of conditions consistently provides assurance of continued process performance and product quality. And therefore, appropriate specifications of the drug substance or the excipient lot-to-lot variations can provide assurance of the state of control. And also for some processes here, incoming material characterization may require multiple physiochemical evaluations in order to cover the failure modes. Now, the next question we want to answer here is, what are the relevant material attributes for continuous manufacturing? And here, I borrowed one of my favorite examples from Dr. Musio's group at Rutgers University. In this case study, they were trying to see how selection of tracers can impact the residence time distribution measured for a continuous mixer. We know that the residence time distribution is an important concept in continuous process because it can tell us how much time the materials stay inside the system. And to experimentally measure the residence time distribution, especially when the amount of API is not sufficiently available, very often we need to use a tracer to track its concentrations over time. And what they found here is that depending on the material properties of the tracer, we can get different residence time distribution. And if we look at the figure to the left, the properties of the tracer is very different from the API. And therefore, the measured residence time distribution is statistically different. As a comparison, if we select a tracer material that has similar properties with the API, like what the figure to the right is showing, then we're more likely to get an RTD that is similar to the API. And I guess what I'm trying to say here is that while continuous manufacturing offers many advantages over traditional batch process, it also brings some challenges. For example, for a robust product and process development, additional material understanding may be needed for certain process modules. And here, let's take a look at the simplified scenario in the continuous direct compression line. For hopper design, the material properties such as bulk density, cohesion can impact the capacity, the outlet size, and the shape of the hopper in order to induce a nice mass flow. For feeders, studies have shown that the initial feed factor, the feeding performance, and the refill deviations are dependent on the material flow properties. And also, as we discussed earlier, the residence time distribution can be used to track the time the material spent inside the system, and therefore can inform material traceability decisions. And in that case, selection of the tracers is important to represent the bulk properties of the in-process materials. And at the tablet press, the flow properties can also impact the weight uniformity and ultimately content uniformity. And that brings us to the next question. What is my material? I myself used to spend in lab working on characterization of bulk solids. So to me, this question is usually followed by a series of sub-questions. For example, how does the material behave under certain conditions? And what are the variabilities of the material? And then, in order to characterize it, what characterization test I need to use? And for that typical test, what test conditions I need to select? During formulation development, how can we select a material? And in the continuous manufacturing, if I want to compare one material to another, what type of materials I need to look at? And how to leverage what I know about the materials to facilitate the process development. Some of these questions have been already discussed in published papers, and some of the questions here might be out of the scope of my presentation today. But with those questions in mind, I would like to highlight some of the case studies going on at FDA from the Division of Product Quality Research in the Office of Testing and Research under the Super Office of Pharmaceutical Quality. Specifically, I will start with one single test, which is the shear cell test. And then I will talk about how we expanded our material knowledge base to multiple testing techniques. Finally, I would like to talk about how we use the material knowledge base with some multivariate analysis to explore it. One of the most commonly used tests to measure flow properties of the material is the shear cell test. There are different types of shear cells that are commercially available, and in order to perform a shear cell test, we need to define several testing parameters. For example, the initial consolidation stress or the normal load at pre-shear, and also the number of the yield points in order to extrapolate the molecular model to get parameters such as cohesion and flow function coefficients. And then we also need to specify the consolidation stress or the normal load for each individual yield point. And like other testings, rushing to pick a random number and start the testing is really not helpful for a meaningful measurement. In Asian China, there is a saying that sharpening your axe will not delay your job of wood cutting. And I think this is particularly true when we want to define testing parameters for a powder flow characterization. The reason we want to characterize powder material is that we want to know how the materials will behave in the manufacturing process. And therefore, in order to have a meaningful measurement, we may want to know what are the conditions of the manufacturing process so that we can try to match the testing conditions with the conditions in the real processes. And luckily, solid processing has been used extensively in other industries, and we can use lessons learned from other industries to help us better characterize materials under more pharmaceutical process-relevant conditions. And here is an example in which we evaluated the consolidation stress inside a feeder hopper. So the one to the left refers to hoppers with vertical walls based on equilibrium of forces on a differential element. The one to the right applies to hoppers with angular walls with the assumption that the radial stress field, in other words, the magnitude of the stress in the converging section of a hopper is assumed to be proportional to the distance of the element from the hopper apex. By calculating the stress conditions inside the hopper, we can then select the appropriate testing conditions for the materials. And using this method, we evaluated the consolidation stress of a loss in weight feeder. And we actually did that for 6 different materials. I like to call this plot a rainbow plot because of the way the consolidation stress at different material field level is colored. For example, the first figure here suggests that for calcium phosphate anhydrous, the consolidation stress is a function of material field level as well as the locations of the material. And the maximum consolidation stress happens when we have the maximum material field level and when we are looking at the bottom of the hopper, which makes sense, right? But what becomes interesting is when we start to compare these with more materials. And we notice that as the feeder keeps delivering the materials and the hopper level is decreasing, the change of the consolidation stress may be different depending on what materials we have. For example, the MCC 102 will only change about 1 kilopascal, but cross-cumulus sodium will change about 2 kilopascal. And also the rate of change is different if we look at how steep the rainbow is. And I think that information captured here can be potentially helpful to evaluate the design space of the feeder or developing the refill strategy of the feeder. We know powder flow is complex and rarely can be described by only one single parameter. And therefore, within our lab capabilities, we started to expand our material knowledge base. In addition to shear cell test, we also started to look at compressibility profile, which suggests the change of the material bulk density under different consolidation stresses. We also measure the permeability profile so that we know how easily the air will pass through the powder bed at different consolidation stresses. We also started to measure the powder flow in terms of the energy it requires to move a blade in an attempt to capture the powder flow when it is dynamically moving and when it is interacting with a blade. Other tests such as particle size and dynamic vapor absorption are also included in our in-house portfolio to support the advanced understanding for continuous manufacturing. And currently, we have 20 materials characterized in our knowledge base, and each material is measured for more than 40 properties. And for those 20 materials, once their properties are measured, we started to explore the database using several multivariate analyses to explore the correlations between them. And there are several ways to do so. And here I'm providing 2 examples showing how to find correlations between different materials. In terms of machine learning, this is part of unsupervised learning. We can either use clustering algorithm to classify materials into different clusters. And in this figure, the material marked with different colors suggests that they are within the same cluster. Or we can use principal component analysis to reduce the dimensions of the data set from 40 variables to 3 variables here. For example, in this figure, materials that are close to each other suggest that they have similar flow properties. And in order to quantify the similarity, we can use similarity metrics such as weighted Euclidean distance to calculate that. And if you still remember the example on selection of tracers to measure the residence time distribution we discussed earlier, and this can be a really good case where we can use multivariate analysis to determine the selection of tracers so that the RTD we measured using the tracer is representative of the RTD of the APIs or the box solids. With data-driven approach, we can also see the correlations between properties from different characterization measurements. And right now, this is based on 20 materials and 40 properties for each material. So the data we are looking at on this slide are roughly 800 data points. I'm certainly aware that this is a data-driven approach, and like other data-driven methods, the correlation we observe doesn't necessarily imply causations. But nonetheless, there is still much information that can be potentially useful. The figure here shows that many of the properties are correlated, and which makes us start to wonder that if many properties are correlated, are all 40 properties necessary to test the material every single time? And if not, is it possible to find and validate a reduced set of measurements so that we can quickly characterize a material with less measurements? And I call this reducing the dimension of measurements. And in order to try to answer this question, we perform a simulation experiment. And this is really inspired by the backward stepwise fitting during the regression. When we try to fit a model with multiple coefficients, we can start with including all the coefficients, and then step by step, try to eliminate one coefficient to see if it passes certain statistical criteria. And if we did the same to reduce the dimension of material measurements, in general, we did it in two phases. In the initial screening phase, we would like to find among 126 scenarios which set of reduced measurements can capture most information. And this stage was done using 15 materials. And then during the validation stage, we try to validate the reduced set of measurements by adding 5 more materials into the data set. In this way, we compared through correlations between 126 scenarios, and we found that there are 12 sets of measurements that can be considered as reduced set of measurements. For example, we can remove dynamic flow test, the shear cell test at 3 kilopascal and 9 kilopascal, where we still capture more than 95% of the information from the original data set. And I just want to mention that the idea of this dimensionality reduction experiment is really not to prioritize one test to another. I'm not saying that the test that can be removed here is not important. The idea here is to prove the fact that many flow properties that we measure are correlated with each other. And because of that, although we didn't do test A, the information may still be captured by test B or test C. As we went through these case studies, you may notice that I used several material characterization tests. However, this doesn't necessarily imply that implementing continuous manufacturing requires tighter or additional material control specifications. However, implementing CM potentially requires additional understanding of the material manufacturability and variabilities for process development. In the end, we need to consider whether the process is robust enough to deal with the variabilities from the material properties so that the quality of the final product is controlled. During the product quality risk assessment, material understanding is important for continuous manufacturing. We have discussed the role of material properties and show that characterizing flow properties of incoming materials can be helpful to evaluate the manufacturability of the drug products. Certain material properties may need to be evaluated for certain unit operations in order to implement the CM technology. Last but not least, control strategy is commonly discussed in CM. And in fact, raw material property evaluation and control is also a key element of the control strategy for continuous manufacturing processes. And finally, I'd like to thank my colleagues and also the leadership team in the Division of Product Quality Research within the Office of Testing and Research under Office of Pharmaceutical Quality. I would also like to thank the FDA's Emerging Technology team. And with that, I will conclude my presentation today, and I will give this back to Stephen. Stephen.
Thank you, Dr. Wang. Once again, I'd like to remind our viewers that there will be a live question and answer session at the end of the presentation, so please feel free to send in your questions at any time during the presentation via the questions tab located at the right-hand side of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second speaker, Mr. Dirk Leister. Mr. Dirk Leister is the head of the application team for extruders and compounders at Thermo Fisher Scientific in Karlsruhe, Germany. The team focuses on applications for extrusion-based processes used in the pharmaceutical, food, and polymer industries. Mr. Leister holds an engineering degree in biotechnology and started his professional career in pharmaceutical research and development. After moving to the instrument manufacturing side of the industry, he had several positions in technical sales and product management. His expertise is developing applications for process equipment used to conduct formulation research, process development, and manufacturing scale-up for pharmaceutical and bulk material handling. Mr. Leister is adept at helping companies minimize optimization time, costs, and efforts of their continuous process scale-up. I will now be handing over to Mr. Leister. Welcome, Mr. Leister.
Thank you, Steve, and thank you for the nice introduction. I'm very grateful to have the chance today to have my presentation after Dr. Wang's nice presentation, and really show the audience a bit of the twin-screw granulation process, which is one of the processes that has found its way into the commercial continuous manufacturing of product, as Mr. Wang was showing at the beginning of our presentation. So what I'd like to outline my presentation is have a look at the continuous manufacturing and its advantages over the batch manufacturing, a look into the twin screw process of granulation. Then very well, as we just learned, there's not only the parameters of the raw materials necessary, but also a sound understanding of the process. So look into the relevant process parameters for TSG and their influence on the process. And everybody from moving from batch to continuous technology at some point in time would ask about the comparability of these different processes. You already know for decades in batches and how they would compare to TSG, for example. And then I would like to conclude with some remarks and case studies on the scale-up of a twin screw granulation process. There have been several publications on the advantages and disadvantages and the challenges of continuous manufacturing. If you look, what I will do throughout my presentation on the twin screw granulation side of the business, these continuous technology helps us tremendously saving in time and material during the development and research of a new formulation. Having instrumentation that is quite small in size and throughput, and being continuous of nature, so we can screen a lot of process variables, and this can help us save us up to 80% of the precious time and the very costly materials involved in the very early R&D stage of a new drug development. Going to the production side of continuous manufacturing can help you to increase your flexibility that we sometimes don't know from batch processes. There is less storage needed because normally we have one train of material go through the dosing until the final drug process. So we don't need any storage area or shipping intermediates around. Typically we have a very small volume at the time which has been processed. So if something goes wrong in a process, we have less waste and don't have to discard a complete batch of material. And being very flexible in setting up the production, we can also react to unforeseen market demands like epidemia in one end of the world, which we didn't forecast 2 years ago. And the quality aspects that we heard quite a lot of, I just wanted to highlight one point here. Because we can equip the process with quite some analytical tools online, as soon as we can ensure a constant process, that means that we also have a constant product quality throughout until the end. As I said, today I will look at the twin screw processes, the twin screw granulation. So the invention of the screw goes back quite in time to dear old Archimedes. And this single screw, as we see here in the animation, has been used for different industries throughout. But if you look at the granulation, that's the point of interest where we have the discussion today. Single screws, they have a limitation in processing because they build up a very, very high pressure. The screw rotates inside the processing chamber, the so-called barrel. And it has a very poor mixing capability, and this is something that we desperately need for the granulation process. So it was until the 1950s where the twin screw extruder was developed, actually as a tool for polymeric chemistry. And the twin screw extruder, in contrast to a single screw sibling, exhibits a very nice mixing capability and does introduce a lot of shear energy that help us achieve the agglomeration that we are going to need for the twin screw granulation process. On that slide, we see three different sized extruders for pharmaceutical granulation that we produce here at Thermo Fisher Scientific. From the left to the right, our Pharma 11, 16, and Pharma 24 granulators. The number in the name of the instrument is for the diameter of the screws running inside the barrel. So we have an 11 millimeter, 60 millimeter, or 24 millimeter screws. You can see that the range of the material flow through these extruders differ from a half kilogram up to 80 kilograms per hour. So are different sizes of extruders available for the different scales you need for development and research. But one thing I would like to point out with continuous manufacturing, in contrast to batch manufacturing, sometimes a small-scale extruder like the Pharma 11 extruder here might be sufficient to be used as a production machine if the time allows and the throughput gives you the material at a certain point in time you need at the end for production. So this is the Pharma 11 extruder a bit more in detail, because the following case studies and data I would like to present was achieved using that model in our laboratory. And as you can see here, the benchtop extruder is for pharmaceutical use, so have a very easy disassembling, assembling, and all product contact parts here just in front of the machine can be disconnected from the drive and measurement unit and can be cleaned, so there is no worries about cross-contamination throughout the whole experiments. So when we look at twin screw processes for pharmaceutical usage, they normally fall into two different categories. One is the extrusion side, which is the left branch of my tree here, so to speak. The extrusion, the hot metal extrusion, is a process that's well known for pharmaceutical usage throughout since the 1980s. And the right branch is the granulation, which splits up into the wet granulation, but also very interestingly, extruder can be used for dry or melt granulation here. But today, I would like to focus on the wet granulation possibilities in pharmaceutical twin screw processes. So here we do have a schematic of an extruder, one for the extrusion, one for the granulation application. I would focus on the right, on the bluish colored twin screw extruder. So normally we see the material feed from the right processing to the left. We have one or two solid streams, hopefully well characterized, as Mrs. Dr. Wang just explained, going into the extruder. Then we have a liquid feed stream as the agglomeration liquid, and then inside the extruder the two screws rotate in the same direction, making the shear energy, the mixing and the kneading, and actually achieve the agglomeration. The end of the extruder when used for granulation is an open discharge. So maybe in contrast to the left picture where you have a die at the end and where you see this gray box indicating a pressure build-up, we are going to release the product here to open air. So this is a pressureless system, and therefore normally with the same extruder you use for extrusion, you have higher throughputs in granulation. Okay. So how do we achieve here the agglomeration? I said this is some close-up photographs and a drawing of one of the two screws is sitting inside the extruder reaction chamber, the so-called barrel, and the indication of the insert of the shear energy that's been achieved through the so-called mixing or kneading elements. You see them in the middle of the pictures, and these elements, I would like to draw your attention to the photographs to the bottom right. These elements can be set up in, first of all, different lengths. So you have all these individual elements stuck together. So you have the different lengths from one element to almost mixing elements throughout your screw design. And another thing is that, as you see to the right-hand picture, the tip of the individual mixing elements, they have an offset, and this offset can vary in the angle between 0, 30, 60, and 90 degrees, as it can be seen here on the picture. And we have a closer look on that one later, because this offset angle gives you the flexibility in introducing shear into your process. As I said in the beginning, we did quite some extensive trials for screening for the process parameters and intrinsic granulation with a Pharma 11 extruder. And throughout the data I'm going to present now, we used placebo formulation. So we started with a pre-blend of 5% PPP, about 30% cornstarch and a bit of lactose, and added a bit of talcum. And as you could see here, water was used as the wettening agent in the wet granulation throughout. Okay, here's a photo of the setup in the laboratory we used. So in the front you see the Pharma 11 extruder. On top of it to the right we have the liquid feeding system, which was an peristaltic pump, so there was no gravimetric control of the liquid feeding. But the solid feeding of the pre-blend materials happened with the means of a gravimetric twin screw feeder inside the barrel of the extruder. And at the end of the extruder we had an Icon 2 particle analyzer from our colleagues of InnoPharma Labs in Ireland to do an online measurement of the granule size, shape, and yeah, a bit of quality control online. So with that testing equipment, we had a look into the influence of process parameters on the actual granule quality. And a lot of parameters like the throughput of the system, the liquid to solid ratio, the screw speed, and the barrel temperature, for example, these are variables one can alter in a set of experiments without stopping the machine. I mean, that's one of the advantages in continuous manufacturing. So you run your equipment, for example, you change the screw speed, you give some time to the system to allow for reaching equilibrium again, then you can have an analyze of your particles. And with that, you don't have to stop your experimental setups and can screen quite a variety of different parameters in one run. Screw configuration, also a very important parameter here. That's been different. For sure, you have to stop your extruder, disassemble the screws, have a different screw configuration, and start all over again. And finally, last but not least, as I said in the beginning, we looked onto the considerations of scaling up to maybe a Pharma 16 or a bigger machine there. So the Icon particle analyzer, which was provided by InnoPharma, is a visual instrument that's been on the end of the process. Takes pictures online and gives you real nice distribution of the particle sizes with a different mean diameters here. And what we could see using a cumulative graph or histograph was that all the measurements we did with the Icon 2 really nicely correlated to sieve analyzers. So it's a bit a different number you might see here, but the effects on the trends is very, very similar to the sieve analyzers you find in granulation quite often. So the first parameter we looked into was the liquid to solid ratio. That's one of the parameters also in the batch granulation that's very important to play with. So the graph to the left is the particle sizes from 0 to 4,000 micrometers. So what we see here is a cumulative curve of the particle distribution, so the volume percentage with a representative mean diameter here. And the different color codes on the graph going from 18 to 28% of liquid into the system. And what one can see is, starting with 80% liquid, is the particle size distribution to the very left. And by increasing the liquids, so we increase the mean diameter of our granules, so the whole graph with the increase of the liquid shifts towards the right. But we could also see that the slope of the graph always stays the same. That means, so we keep the same particle size distribution, but we just shift around the mean particle size diameter. With increasing of liquid, we see an increase in oversized particles. That's been due to the better agglomeration here. We see a decrease in the fines because we have a more thorough granulation overall. And what can also be seen, that was something we didn't measure, but that's been noted in literature, that we have an increase of density of our granules. The right graph shows you the different D50, D90, D10 values, and that really goes into line with what I just said, that we stay always with the same particle size distribution around increasing mean particle size here. So what happens if we use the extruder to vary one parameter, so the screw speed here? If you remember the setup of the screws I showed in the beginning, so we have conveying elements pushing the powder and the wetted powder through the system, and the mixing elements. So the number of mixing elements, as we are going to see, is far lower than the one of conveying elements. So if I increase my screw speed, I increase the shear energy, and I normally chop off my agglomerates, so getting fine agglomerates. And this is what we see in the graph here. From 300 to 900 RPM, we kept the throughput constant, about 1 kilogram per hour. And by increasing the screw speed, so we could decrease the average particle size. But as you can see here, in a certain range, 500 to 800 RPM, there is not that much happening in respect, but lowering our screw speed gives us really, really big agglomerates here. And this also goes very nicely in line of what we found in the literature. So, and as you said in the beginning, the mixing, the shear energy we introduce into the system is quite important for processing our agglomerates. And there is quite some things we can do about increasing our mixing capabilities here. First of all, the amount of kneading elements and the offset angle of the kneading elements, as I said, or as these three pictures in the middle of the slide presents, we have to the left the conveying elements. They transport the powder through the barrel, but because we have a small clearance to the wall, there is still already some shear energy we put into the system, and there's also some literature published using only these for agglomeration. Then in the middle we have so-called distributive elements. They have a bit higher shearing, a better mixing. And to the right, that's the already known mixing elements with a different offset angle between the individual elements. And the more I mix, the more I form agglomerates, and the bigger the mean diameter of my particles will get. Okay, I will go forward to that slide here just to give one, let's say, rule of thumb. So the conveying capability and the mixing capability of the individual elements. So the feed screws or the conveying screws on the top, they, as the name implies, transport the material throughout the system, and they have a very, very small shear energy to introduce, so their mixing capabilities are quite low. If I have mixing elements, their tip has an offset of 30 degrees angle. The conveying capabilities, they drop down, but the mixing and the shear energy is intensified. 60 degrees, 90 degrees is the same. So the conveying capabilities of these mixing elements drop down until down to zero actually. And the mixing energy, the shear energy we put into the system is the highest here. So at the 90 degrees alternating mixing elements, if you stop your extruder or you stop your feeder running the extruder empty, you will always find some powder remaining in the system here. So the barrel chamber and the volume we process is quite small. So if we do a lot of mixing elements in one spot, and if we do a lot of intensified mixing in one spot, there might be a risk of overheating your material. So strategy to avoid that is depicted here in that picture. So we have a screw section of the barrel and 4 different screw setups. So the material flow goes from the right to the left. So you see the feed port opening for the powder material on the right. And then down the stream, you see the liquid port injection here. So the first screw setup is a screw with only conveying elements. As I said in the beginning, depending on your formulation, you can use that for doing some mixing, but it's a very mild, no intensive mixing. And then the following 3 screw setups have mixing sections with mixing elements with a 30 degrees offset angle towards the end of the screw. That's the first from the top. Then the second one, and then the third one, always moving more towards the powder inlet. And by distributing your mixing zones throughout the whole screw, you can avoid small mixing zones. Sorry, you can avoid overheating in one spot. So with the different process parameters of the trin screw granulation, the screw speed, the throughput, liquid to solid ratio, and the configuration at least, you can really tailor your particle size distribution. You can tailor your granule density and also the shape and the structure of your granules. And ultimately, with these properties in hand and the knowledge of the raw material identification, you can really design your product quality, which ultimately leads to the desired drug release profile or tablet hardness with your end product. There's also some literature out there comparing the trin screw granulation and the high shear granulator. The later is one of the most popular batch granulation systems used. So the granular quality you get out of the 2 systems are quite similar in terms of density and shape. And both of them are very well controlled and have quite short residence times. What you will find in the literature quite often that with a trin screw granulator you might have multimodal particle size distribution. So instead of 1 average, you have 2 Gaussian curves here, and the high shear granulator has been sometimes considered as having a wider design space, more possibilities. My colleague, Dr. Maggie Richter, which is our pharmaceutical application scientist, did quite a lot investigation in this one, and she found that the multimodal distribution often occurs if you have a pulsation in the material inlet in the system. Might be the feeding of your powders, or more often it's the feeding of your liquid into the system. So if we do have a pulsation here, then we also have a pulsation in your granule quality, and that leads to the multimodal distribution. And as soon as you start using your screw configuration with all the different elements and all the different positions to put in, as I just showed in the slides before, you have a huge design space using one extruder. So I can only encourage everybody doing trin screw granulation to change the screw setup from time to time. Now in my last part to the presentation, I would like to give some thoughts about scale up. Sometimes if you start in a small scale extruder, for what reason ever, might be the availability of the amount of materials at the beginning, you have to come to a bigger extruder towards production. Everything is based around geometrical similarity of the screws. So what we see here is a cross-section of the trin screws, and all these different parameters here, like the auto screw diameter, the channel depth, the center line. This is something we need to keep constant from a small scale to big scale extruder in order to make a successful scale up. So for example, the whole length ratio to the diameter should be constant in each size. The inner to the outer diameter should be constant in each size, and as well the center line and the outer diameter, this ratio should also be constant. So if we keep that constant, we have a geometrical similarities, and all the physical interactions I'm now going to explain hold true in all the scales. For sure, if we go from a small to a big scale extruder, we should also keep the screw configuration the same. There have been quite a number of publications for the scale up. So Schuler in the 90s did one. So the throughput of our production extruder, that's the throughput of the laboratory extruder to the power of 3 with a ratio of the diameters here. So that's an assumption, a good start. So if you go to a bigger size extruder, that's the throughput increase you could expect. The other parameter we should look at is the specific energy, because we put energy into the system, shear energy, we do the mixing, and the specific energy is the shear energy, mechanical energy we put into the system per volume of powder here, so that should be kept constant. And last but not least, we do have the powder flow number, and I don't going to explain that, but in the red box you see in the end, that's the powder flow number is attribute for the filling level in your extruder. I mean, you can have one extruder with a very, very low amount of powder, so you set your powder feeder to a very low amount, or you can have the same extruder with almost the highest possible load in the extruder. But the filling of the screws have to have a certain amount. If we have too less material in the system, that can behave very strangely, and you never can come to a stable process. And this is what my colleague showed here. So we had our Pharma 16 twin screw extruder, open discharge, and we used the UV light. You see the picture to the left, a UV tracer. And we played a bit with the throughput. That's the graph we see below. And with the screw speed. So with 15 kg an hour, increasing the screw speed between 0 to 1000 RPM, you see the line dropping, and that's what we expected. So the mean residence time is faster the faster you go with the screw speed. That holds true for 10 kg and 5 kg, but if you go to 3 kg for this size of extruder, a very low filling level, all of a sudden increasing the screw speed gives you a dramatic increase in the residence time as well. So the filling level of the machine is not high enough anymore. There is no powder enough to push the other powder through the system, so we get back mixing effects, and all of a sudden a very unstable system. So the point I wanted to make here, there's different sizes of extruders for a very good reason, because if you underfill very big extruders, you might have a hard time to reach a stable process. All of the measurements I showed here were for wet granules. So we also did some case studies here after granulation from our 11mm extruder. You see the picture top right, which is a laboratory setup for continuous manufacturing, which would require some manual intervention, down to the complete automated and software-controlled 60mm continuous line until the final tablet press. But also here with the sieve analyzers, the particle size distribution really matches from the experiments we did on the 11 and the 60mm extruder, using just the same scale-up parameters as I was explaining. With this one, I would like to come to the conclusion of my talk. So as we saw with Dr. Wang, it's very important to know about the probabilities of your material goes into, but also the process parameters helps you to design the product you need in the end. So I hope I could show you that intrinsic granulation is a very versatile technology for your different formulation requirements, and that if you start into CM using a twin screw extruder, you can expect very comparable results to other technologies you might be used to. And the granule quality on a small scale extruder is very predictive to what you can expect in the larger systems. Yeah, there's a reference list, I think, for you, and with this one I would like to thank you for your attention.
Thank you, Mr. Leister. Now we will begin the question and answer segment of the webinar. Once again, I would like to remind the audience that you can still send your questions via the questions tab on the right-hand side of your screen. Now, for our first question, how are the materials selected in your material database?
We actually spent quite a amount of time to select the materials in order to include in the knowledge base. We look at the 4 approved drug products currently, and based on the label claim, we select the excipients, and then we also started research the literature published in which some of the materials are commonly used in the experiments reported in the papers. And also we try to cover a wide range of properties, not only for the excipients, but also for the model APIs. That's why we included some of the APIs as well.
And our next question is, are material flow properties always necessary for continuous process development?
I think it depends on what, if you are asking for regulatory requirements. Currently, there is no current requirement for what data you need to submit to report the flow properties. But for process development, I would say most likely you have to look at the properties of the material, especially the flow of the flow properties of the material, because in the continuous process, the process is heavily relying on the continuous flow of the materials.
Thank you. What has to be considered when developing a TSG process?
Okay, I think that one goes to me, Steve. Yeah, I think the screw design, I went through that in my slides, is a feature that is very often overlooked. So if you read a lot of publications, screw design is somewhat changed, not that often as other parameters. So that's definitely something I would consider when designing a TSG process. Very naturally, if one comes from, for example, the high shear granulation, transferring the knowledge from there to the extruder one-to-one is a good starting point. But sometimes it's also to see that, for example, a binder is a big point here. In the trin-screw extruder, we could observe that it's quite a difference if you have the binder dissolved in the liquid and put it into the system, or you put it in your pre-blend. So there's sometimes things you might want to consider different in the batch sizes. And last but not least, one point I would like to remember or remind is that there is a scale-up possible in the trin-screw granulation, but sometimes if you look at the final amount of material needed and you look at the time you have at your disposal to get there, you might get away with a small scale or mid-scale sized extruder as your production machine, and that's, I think.
Another question on the back of that one, is scale-up always necessary when going to commercial manufacturing?
Yeah, that's a good one. As I just said, if you look at the amount of material you need at the end and the time at your disposal, you don't necessarily have to do scale up all the time. So it is perfectly feasible, as shown in the presentation here. But we do also have quite a lot of customers running with laboratory-sized extruder and doing a perfect production scale. And if there is a bigger throughput necessary, let's say if you look over the lifetime of your product, it's not always necessary to go the scale-up route, so scaling up to a bigger instrument. But continuous manufacturing also inherits the possibility to scale out. That means you duplicate your granulation extruder for a certain time. You need the capacity to double, and you don't have to really scale up your process. And if the lifespan of your product doesn't require that throughput anymore, the second extruder has been free doing some other trials or process development work.
Thank you. I'd like to open up the floor now, so both presenters who are able to answer these next questions. Can we facilitate online in-process testing of product?
Of course we can. I guess you're asking about how we can facilitate the in-process testing of the product. Maybe you can take advantage of the emerging PAT tools. And also, if I can go back to the presentation I presented, the in-process testing always starts with your understanding, both for the materials and for the processes.
Yeah.
Thank you.
I mean, we do quite a lot of online testing or online measurement throughout the R&D and the testing, and as Mrs. Wang said, there's quite a lot of technologies emerging. So I like visual instruments very much, but also the NIR or microwave detection of moisture, for example. That's really interesting and a good PAT tool to control your process.
Thank you. Next question. This one is for Dr. Wang. Apart from the mixture RTD difference, is there observation on any change in final granule attributes from twin screw granulator that is affected by material properties?
Oh, thank you for the question. Actually, in this case, we didn't go further to wet granulation. It's a direct compression case, and we are focusing on the RTD of the mixture itself. But I think you bring a very good point. Sometimes we show there is a difference in RTD, but depending on your specifications and your the RTD itself, you may not see the significant difference in the content uniformity. So one way to see it would be interesting to combine the RTD difference with the funnel plot to see what you will get in the final product.
Thank you. And Mr. Leister, if you do have any thoughts, would you like to voice them? So that's, apart from the mixture RTD difference, is there observation on any change in final granule attributes from twin screw granulator that is affected by material properties?
I have to give thought on that one, just a minute. So
no problem. We can come back to that question.
Yeah, we can. Better that one. Yeah.
Our next question is: cleaning validation for CM is challenged. They'd like to listen to your views on this. So I'll say that one again. Cleaning validation for CM is challenging. Do you have any views on this?
Yeah, I can maybe start with this. Thanks for the question. I think you bring a very good point, because previously I talked about process development, control strategy, and material properties. The cleaning actually falls into another category, which is the facility operational aspect, or the GMP compliance. It really depends case by case, but in general, some things to consider might be your sampling conditions. Is it sufficient and representative? And also the hold times, especially when you're talking about wet granulation. And also other things to consider might be to evaluate your hold time for the dirty locations, and also the sampling test for the visual inspections, as well as API residues or the micro monitoring. Yeah, that's something in my mind.
Yeah.
Our next question is, an audience member needs to understand the RM testing through PAT tools, i.e. RM testing at warehouse or before start of execution / at the time of execution. Remember, this is both of presenters.
Raw material.
Yeah, raw material testing.
Mm-hmm.
So for raw materials, you usually have the certificate of analysis, and for continuous manufacturing, if you are adding additional characterization, it's very often part of your internal quality management system. You are asking if it's tested at warehouse or before the execution or at the time of execution. If your material are stored in a controlled environment, they're supposed to be the same, right? Does this question imply the material properties will change? I guess I'll bring that question to you.
Yeah, I think the material properties I would think of from having in my mind continuous extrusion example is, for example, the if you have a huge or quite big feeding systems with a feed hopper, you showed that example of the bulk material density. So the likelihood of bridge building that can change with the time the material is sitting in the hopper. So this is one raw material attribute you might test beforehand, but that has maybe significant change through a 24/7 operation here. So the testing happens at the execution, happens at the process. So this is something of what we've been going through with some partners where we did long-term trials of a learning curve. So the material tested, specification was okay, but the physical properties sitting in a hopper for 24, 48, 72 hours can change dramatically for the bridge building behavior, for example. So there's quite some points you would like to consider in a continuous manufacturing you might not consider in your testing, in your standard testing procedures.
Thank you. And with that comes to the conclusion of the question and answer session. Thank you for everyone who answered questions, and unfortunately we can't go through them all. And before I finish the webinar, I would like to ask our presenters, Dr. Yifan Wang and Mr. Dirk Leister, if they do have any closing remarks.
I would like to thank my colleagues as well as my leadership team. I also want to thank Biopharm Asia and Stephen to organize this webinar. Thank you.
Yeah, I can also say thank you to Biopharm Asia and to you, Stephen, for hosting us and giving us the possibility to share with the community our thoughts. And I also would like to thank especially my colleague, Dr. Richter, and also the colleagues from InnoPharm Labs, Chris O'Callaghan here by name, for the supporting of the trials and the data I could present throughout the presentation here. Thank you.
Well, thank you both. I'd like to take this opportunity to thank our presenters once again, Dr. Yifan Wang and Mr. Dirk Leister, for sharing their knowledge with us. I would also like to remind our audience that they can view this webinar and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. Thank you, everyone, and have a great day.